Well, good morning, everyone. Thanks for attending. I'm going to be just talking about a slightly different approach to cell therapy with the approach that Notch has been taking. There's an old saying about you should really start with the end in mind. And Notch was founded on the belief that to improve cellular therapy, there we go. That the real benefit of cell therapy today is in the autologous setting, where we take a patient's own blood cells and derive them and manufacture them and re-administer them to the patient. And today we're seeing great advances in improving the manufacturing of those cells, but still it's always going to be a one-for-one personalized medicine, and the health of the patient, the health of their cells is always going to be variable from treatment to treatment. And so the long-term goal in allogeneic cell therapy is, of course, to be able to have a universal off-the-shelf approach. A number of companies have embarked upon this. Notch was founded in 2018, and the whole goal was, how can we really turn the creation of cells from pluripotent stem cells into therapeutic T cells in a uniform, highly scalable, produced way. And so today I'm going to just talk a little bit about what we've achieved and where we're going as a company. But I would say today we believe that our data supports that we have proven that we can have unprecedented production of T cells from pluripotent stem cells in a highly controlled manufacturing system that is really a small footprint. And as as we heard in today's announcement from Bayer, their large production facility in Berkeley. And if we look at many of the companies involved in this space, very large amounts of money being invested into the infrastructure required for manufacturing. And we're hoping to change that, to be able to move to small footprint manufacturing that would be very much akin to what is used in production of antibodies or other proteins. So I don't expect you to read all the detail here, but this is really a schematic showing how you would start from a pluripotent stem cell, transform that stem cell into a master cell bank of first CD34 cells. We've transformed this all into stirred tank reactor technology, and that becomes our starting material for then our proprietary process of how to go from CD34s through to a single positive alpha beta CD8 cells. That last step in the drug product manufacturing, the differentiation of a CD34 to T cells, has proven to be very problematic for others in the field. And it really comes down to why is Notch called Notch Therapeutics, and that's because of one of our co-founders, Dr. Juan Carlos Zuniga-Fluker, very early demonstrated that triggering the Notch pathway is critical to causing pluripotent stem cells to differentiate and commit to the T cell lineage. And so we believe that we are the experts in Notch signaling, and we have developed a proprietary way to do this in a scalable fashion. And the fundamental aspects of our intellectual property are the use of magnetic beads coated with Notch ligands, with DLL4 as well as VCAM that have been shown to be synergistic signaling partners to very precisely control the trajectory of stem cells through to T cells. Now, the real advantage here is not only can you control the signaling, the strength, the duration, but now that we're using a bead-based format, we can move into a three-dimensional suspension-based process, which now has volumetric scaling, which, of course, from a cost of goods perspective, is very appealing. But it's also now we're working within a closed system where we can also control the feeding rate for these cells, the whole oxygenation state. So many parameters that when you think about now from an FDA perspective, this is really what we're trying to move towards, is a way to make cells in a way that will be highly controlled and creating a well-characterized product. Again, very much like what you would expect for an antibody or a protein. And this is what's really been missing in cell therapy. The former commissioner, Dr. Scott Gottlieb from the FDA, has been in many talks, and he's said repeatedly that the problem today in cell therapy is we can't really tell if the variability that we see in the clinic is due to the variability in the biology or the variability in the manufacturing process. And we seek to change that. And so I always like to highlight that this isn't just about a manufacturing process and a cost of goods, though we think that that will be a very important step in improving the accessibility of patients. But now we're talking about cells that are uniformly made so that we can have, a large supply of product for many types of clinical investigation. And so it's really the benefit that we will bring to the clinical investigation of cell therapy to be able to show that, what is a dose response? Does anybody even realize that in cell therapy today there really has not even been a good dose response study done because it's really not practical with autologous cell therapy? So this is something that we hope to change to make cell therapy more drug-like. And then this should ultimately translate into better clinical results and better outcomes for patients. So today, again, I think we've done a very good job of showing that our process can create the cells we want. They have proliferative capacity, just like a primary T cell. And I say just like a primary T cell because our philosophy within the company is that we're not trying to absolutely replicate a primary T cell. I don't think we're ever going to be able to do what the thymus does, but we think that very phenotypically and functionally, our cells are behaving very much like a T cell should be, and they have the markers that we would want them to have. And then from a scalability, we're already working in bioreactors that we need, and the potency of these cells in vitro has been showing to be very much like primary T cells, and I think you'll be seeing from us very exciting in vivo data very shortly. So this is, again, just, when we talk about what is transforming in cell therapy today, again, within autologous, we're seeing these closed manufacturing systems making huge advantages over what was previously used in autologous cell therapy, but still the supply chain, patient collection logistics, having to have lentivirus for transfection for every new batch. This, again, supply chain and the cost and the facilities are still quite expansive. And the goal has always been able to move to allogeneic cell therapy to make this therapy more widely known, or widely available. But even what we're seeing in the healthy donor sector, again, the supply chain logistics are quite daunting, and the manufacturing costs are not coming down as greatly as expected. So we hope to be at that far right end of pluripotent stem cells, where you can make a clone, select that clone, that now becomes your universal starting material in what we see as a very small footprint manufacturing process. We're now doing 1 liter bioreactors, creating billions of cells in a 1,000 square foot room with 2 operators. Again, that's kind of unheard of in this business. So I gave you a lot of claims without showing you a lot of data. That's why I've got this up here. All of our data is on our website. You can see that we've had a number of very important recent publications outlining some of this data that I've alluded to. Also, we've got really important data coming at SITC. We'll likely also be at ASH. So we always encourage everybody to go to the source, go to the real published data to see what I've been talking about. But as a company, where we are today is we think we've created this commercial-ready allogeneic cell chassis that we can manufacture at scale using our bioreactor-based manufacturing. And now we are starting to work with partners on inserting CARs and TCR. And all of the examples I was giving, we've been able to do this with both CAR-based insertion into these cells as well as TCR. So we think we have a broad applicability to a number of different products. And so we're also, of course, interested in talking to more partners. If you have your favorite CAR or TCR and you would like to move towards an allogeneic cell therapy, we'd love to talk, as well as Notch has been in its own process of acquiring its own binders for our own pipeline, and we're starting to advance those as well. So hopefully I've outlined where Notch is today, that I think that we can fully enable iPSC-based T-cell therapies. One thing that I skipped over was, again, everything has been created in a stir tank reactor, and CD34s are a very important starting point in our differentiation platform. So we now are also, I think, one of the world's leading potential suppliers of CD34. So if people would like to move away from cord blood-derived CD34s or donor-derived, that's something else we'd be also interested in talking about. So we're really, as a company, in our own product pipeline, focused on the CD8 alpha-beta T cells. We think, again, we are the only company in the world that's been able to show consistently able to control that process to get those kinds of cells, and that we're now, advancing those from an in vivo perspective, getting ready for IND-enabling studies. I think I've got a couple minutes left if there's any questions. Is there somebody back there? Yes.
If you add a TCR back to your T cell, it will cause the GVHD.
Pardon me?
If you add a TCR back to your T cell, it will cause the GVHD.
No, that's, we first eliminate the intrinsic TCR, we knock that out, and the TCR we're putting back in is very specific for a particular antigen that it's selected for, for a cancer epitope.
Okay, thank you.
Yes, sir.
How do you scale up the production of your iPSCs while the process?
There's a lot of background noise. If you could just speak up a bit.
Here, hold on. This is soft, you can catch it.
Excellent.
How do you scale up the part of your iPSC production? So you've got iPSCs and then you've got a differentiation in stirred tank reactors.
So how do we start it off?
Yeah, how do you produce enough iPSCs to then turn them into,
Great, yeah, so that's, that does not require the bead system that I talked about. That's really for the end stage. So at the beginning, some of our expertise, our Chief Technical Officer, Emily Titus, that was her background, both in reprogramming mature cells back to iPSCs, and then the whole scale-up of using aggregates to be able to get them to CD34s. So we do all that in stirred tank reactors as well, but does not require the bead-based technology. In fact, we have more CD34s than we know what to do with. That's one of the benefit of that process. Well, thank you very much for your time, and I'll be around if anybody wants to talk further. Thank you.